Well, it gives me a great pleasure to introduce today's speaker, Dr. Taha Merghoub. He's the Deputy Director of the Sandra and Edward Meyer Cancer Center at Weill Cornell Medicine, and congratulations, Taha, on that appointment. He's the Margaret and Herman Sokol Professor of Oncology Research and a Professor of Pharmacology and Immunology Research in Medicine at Weill Cornell. He is an esteemed colleague in the field of myeloid research. And I know I've known him for quite a while when we started looking at PI3 kinase gamma inhibition in solid tumors. And we published papers at the same time in Nature, and that led to the development of a drug. So he received his BA from the University of Algiers in Algeria, and then MS and PhD degrees with the highest distinction from the University of Paris in France. He then went to do postdoctoral research with Pier Paolo Pandolfini at Memorial Sloan Kettering Cancer Center. He then joined the Allen Houghton Lab as a research scientist to study tumor immunology, and began a research laboratory in partnership with Dr. Jed Wolchuk at Memorial Sloan Kettering, became a full member, and then now has transitioned to be the Deputy Director of the Cancer Center at Weill Cornell. He studies immune responses in multiple cancers and has done terrific work on examining myeloid cells and molecular mechanisms of myeloid cells and other cells in solid tumors. So today he'll talk to us about innate cells as effectors of immune escape variants, elimination, and immune adverse events. So welcome, Taha, and thank you.
Yeah, thank you, Judith, and thank you, Jennifer, and the whole group for inviting me in the Myeloid Network. I feel very connected now. I am going to share my slides. You can see them, right? Are you seeing the right view?
Yes.
Yeah.
Perfect.
So, so as Judith said, I'll tell you today about some of our serendipitous observation or discoveries that we've made by looking at something completely different, and I'll tell you how we got there. And so those are my disclosures. And so our lab, if we are very unfocused, so to speak, so if put in one sentence, we study mechanism and approaches to overcome resistance to immune therapy. We use melanoma as a model system because we think we have the right tools to study melanoma and really being able to look at this in the progressive phases of melanoma progression. Typically, we look at using combination therapies such as immune modulation, vaccine, etc., in order to improve responses. And we like to do translational work, meaning going from the preclinical model to the human and back. And this is generally the theme of the work in the lab. And this is, I know, not stranger to this audience, but just framing, the type of work we do, so you know me better at the same time. We really, and when thinking of tumor evolution from elimination to escape, we are actually interested in reversing this elimination phase by looking at the different mechanism within the tumor microenvironment. And of course, one of them is overcoming, myeloid suppressor cells of macrophages within the tumor microenvironment in order to have a more potent anti-tumor immunity. And so, again, in the lab, we have different folks studying a way to remove immune suppression by looking at myeloid Treg and IDO, boosting existing T-cell responses. We like co-stimulation, and although a lot of people have given up on GITR and OX40, we still think those are great pathways to look at, and we like to study them. We try to, another approach is forcing antigen presentation and what I call supplementing the T-cell repertoire by either immunization, in situ vaccine, or adoptive T-cell therapies. And we're looking at some CARs at the moment. And so the really story started by looking at means to boost existing T-cell responses. And so we were interested in co-stimulatory molecules. And of course, we know that CTLA-4, PD-1, and LAG-3 are now approved in melanoma treatment, and of course CTLA-4 and PD-1 are approved in more than just melanoma. But we were interested in OX40, GITR, and CD40 to a certain extent. And so by looking at OX40 with Daniel Hirschhorn in the lab, we were very interested because OX40 actually increases proliferation and clonal expansion of CD4 effector cells. It does something similar in CD8 effector cells when engaging them and agonizing these cells. But very interestingly, when you engage CD4 Tregs, then you deactivate their function. And we believe that there are multiple mechanisms by which this is happening, and some of it is also by elimination of these cells by AICD or through FC receptor. And so what we had seen initially is that engagement of OX40 as a monotherapy is ineffective in treating established tumors. So it worked as a single agent when we had smaller tumor, but it didn't work as a single agent when tumor were larger. And so if somebody's familiar with how B16 are aggressive, you see that we were able to treat large tumors, which are at least half centimeter, quite big tumors, by giving them a regimen of 1 dose of cyclophosphamide. And the belief is that when you give cyclophosphamide, in this case, you engender lymphodepletion, which allows for homeostatic proliferation on the antigen-presenting cell, not on the T cells, sorry, which come up in the presence of antigen release. And the antigen release is because the B16 will be sensitive to a certain extent to cyclophosphamide. And what we did one day after this regimen is adoptively transferring CD4 antigen-specific T cells, which recognize TRP1, which is a melanosomal antigen, on tumor cells, in addition to OX40 agonist antibody. And what we saw is that after 3 weeks, you have complete regression of these tumors. And this is a quite potent treatment regimen. And so this was, depending on where you are, I think this may be close to lunch or after breakfast. So I hope this does not make any effects on you seeing these gross images. But we use the metabotropic receptor model that Susie Chen has developed at Rutgers, where deregulation of the GRM1, which is the glutamate metabotropic receptor, will lead to melanoma formation in mice. And what's interesting is that in this GEM model, when untreated, we really have nasty tumors showing in a non-haired skin, because the melanocytes are in the dermis in this animal, in furry animals, in the area where there is fur, that mainly in the hair follicle. And so, and you can see that when you use the combination therapy, these mice would, cure their aggressive tumors, and at the same time, you have certain degree of depigmentation, which would be equivalent of vitiligo in patients. And so, and what we saw also, which is not something we expected at the same time, is that we had this in the ears or in the snout and the tails, you had this skin effect where and in the ears, we call this cauliflower-like ear, which is seen also in mice where you deplete Tregs, and actually this sent us in the wrong direction because we're mainly thinking about Tregs initially. And the nice thing is that you can actually measure ear thickness as a surrogate marker of these side effects in these mice. And so, and then we wanted to actually tackle these questions of tumor heterogeneity. And the question that we asked is, when we target a single antigen, if tumors are heterogeneous, is targeting a single or a subset of antigen an effective strategy? And if so, do we need to target every clone for tumor elimination? And can we mobilize endogenous immune system to eliminate heterogeneous tumors? And when we're thinking of mobilizing endogenous immune system, we were thinking about the adaptive immune system. We were not thinking of the innate immune system. And so basically, right, I mean, I think this is clear to most, but so if we have a homogeneous tumor, if we treat for TRP1, for example, or a given selective pressure, then we eliminate everything. However, if we have subset of cells which do not express the antigen or do not express whatever the selective pressure we put on those tumors, then we have, a persistent growth of the tumor, and the persistent clones will actually take over the tumors. And so in trying to see how we can model this, we looked at patients, and so those are different sections from melanoma patients. And you see, for example, if we looked at TYRP1 as this antigen, you see that some patients will have in their lesion high expression of TYRP1, some of them will have lower expression, and some of them will have no expression at all, where the tumors will be completely depigmented. And looking at other data, this is a paper from Meyer, where they did single cell analysis, and we looked at different melanoma differentiation antigen and cancer testis antigen. You see that the expression of these antigens is quite heterogeneous. And here, for example, we looked at TYRP1 in different patients, and you see here again that some patients will express more homogeneous level of TYRP1 and others would not. And so showing that really there is this heterogeneity in these tumors. And so we tried to model that. And how did we model that? We implanted either B16, which are TYRP1 positive, or B7H1 cells, which are TYRP1 negative cells. They're B16, which are mutated, but don't express only, so they're not just not expressing TRP1, they're also TAP1 deficient. So they don't present class 1 or class 2. And what we have shown in the past is that if you implant B16, which is antigen competent, then you see that you have complete regression of the tumor. Now, if you implant the negative tumors, then you see that they don't regress the tumor. However, interestingly, if we put each tumor in every flank, you see that the B16 tumor regresses, the TRP1 negative tumor does not regress. However, if we mix those tumors together, you see that everything regresses, suggesting that, and the expectation is that the B7H1 cells should, grow, and based on the model I showed you, and in fact, it was not the case, suggesting that maybe there is a bystander killing effect. And so what we did by actually following the adaptively transferred T cells in this model, we see that while T cells come in and go quite rapidly, there is a clear peak at the given moment. And we see that tumor regression continues even when T cells are gone and they're not there anymore. And so, and then we ask the question, is it completely T cell dependent? So what we did is we depleted the effector CD4 T cells either early after adaptive T cell transfer of the cells or late. And we see that when we deplete the cells late, you still have complete regression, suggesting that maybe the continuous regression of these T cells is not solely driven on the adaptively transferred T cells, maybe by endogenous T cells which are there or other cells. And so, of course, again, we were very focused on T cells, but then when we tried to look at cytokine profile from tumor extract, we saw that there was that there were many cytokines which are reminiscent of a myeloid component, which were important in these tumors. And so we assumed then we were dealing with the wrong tool. And so when we tried to characterize these tumors more and looking at myeloid compartment, we actually looked at anti-CTLA-4 and OX40 in this case, and you'll know why later. You see that there is there is an accumulation of CD11b positive cells in both anti-CTLA4 and anti-OX40 treated tumors, and they are otherwise 6G positive, and so suggesting that you have heavy neutrophil recruitment in these tumors, and so then we actually started scratching our head, looking at the neutrophil literature, and I mean, at least to me, I found it quite confusing, but we started talking with Michaela, and we were thinking about, I mean, one of the first hypotheses we were thinking is maybe there is netosis, and maybe the mechanism that's happening is associated with netosis, which is the presence of this nuclear membrane and extracellular traps, and so we learn from Michaela how to actually stain for these nets, and I think we convinced ourselves that the treated mice actually had accumulation of nets, and in a quite clear manner, and we tried to look also at the anti-CTLA4 treated mice, and we saw the same accumulation of neutrophils and nets in this treated animal. And so then the question was, is this functional or not? So what actually Daniel did is deplete neutrophils, and if we deplete neutrophil in a homogeneous tumors, meaning the B16 without the antigen loss variant, you don't see much effect. However, if you do that in the setting where you have a heterogeneous tumor, you see that you lose some of the protective effect, and the B70H1 cells now are no longer eliminated, suggesting that maybe neutrophil have a role in the elimination of the antigen loss variant. And so, of course, we were all thinking about nets as being the mechanism by which this antigen loss variant were eliminated. So we did experiment in PAD4 knockout, in elastase knockout, and we didn't see any nothing happened, basically suggesting that net didn't have any effect. And we tried some experiment in vitro, by using DNAs and also using the PAD4 knockouts, and we haven't seen anything. However, when we actually did this in iNOS knockout mice, we saw that we lost some of these effects, suggesting that in part this is driven by iNOS in these neutrophils, and of course this is a full knockout, and we were not necessarily convinced that this was the driving mechanism, and then so we did some killing, in vitro killing experiment where we took the B7H1 cells and put them in the presence of neutrophils coming from wild-type mice or iNOS knockout mice, and we see there is a clear difference in the killing capability of those neutrophils. And so then with Daniel, we tried to understand why this was the case, right? And so we did what everyone does, bulk RNA-seq, and we see that clearly the mice which were treated with anti-OX40, or the tumor from these mice, clustered completely differently from the IgG tumors, and then to understand that better, we did single-cell RNA-seq, and we actually very interestingly saw that in fact there was this, there were multiple clusters, of course, and there was this cluster 1, which was associated with the elimination of the antigen loss variants. And so in characterizing this further, we actually saw that, so cluster 0 was the one that was mainly associated with the non-responding tumors, and cluster 1 was more abundant in this more more effective in terms of killing, and so you can see that probably the level of maturity of the killer neutrophil versus the non-killer ones is quite different. And then when we looked at their gene profiles and expression, we see that they have features of neutrophil migration, neutrophil chemotaxis, response to interferon, etc. And so suggesting that really this maybe signature that or this cluster that we are identifying has some role in the tumor elimination of the antigen loss variant that we've described in this model. And you can see here this cluster 1 has chemotaxis capabilities, neutrophil killing, apoptosis, also mitosis, which is associated somehow with this phenotype we see, and you see that the cluster 0 is more azurophilic and expresses gelatinase, and so so with David Redmond, he actually did this trajectory analysis, helping us understand and maybe hypothesize whether these neutrophils are in sequence, meaning that probably one coming from another, or that are evolving differently, probably when looking at the trajectory. And we see that probably the more the cluster 1 is constituted of more mature neutrophils and will express genes such as CBP alpha, CCL4, CL3, FOS, JUN, and IL-1 beta, for example, and 6CL2. And so, and then we asked ourselves, right, what is the translational potential of these anti-tumor neutrophils that we think we have identified? And so we try to, although I'm not a big fan of this type of analysis, I think it still can give us a clue and guide us. So we applied this signature of cluster 1 versus cluster 0 in patient from the TCGA. And what we see is that the cluster 0, which was not associated with tumor elimination, did not actually correlate with anything when we looked at tumor survival. However, when we used cluster 1 in the TCGA, cluster 1 seemed to be associated with patients which have longer term survival. And we took advantage of a phase 1 trial that was done led by Andy Weinberg, or initiated by Andy Weinberg, in Portland, and where they used OX40 as a single agent in this case, and what we see is that and they had biopsies pre and post, and we had access to these biopsies, which was great, and so we were able to look at the netosis in this case as a marker of this neutrophil activation, and you see that in fact, post-treatment, you have multiple patients which develop this netosis. And so, and then the question that we asked, and actually a reviewer asked, not we did, but we asked whether this would apply in other setting outside of the adoptive transfer therapy. And so Sadhna used the MC38 model, where she depleted with anti-LOS6G, which we think mainly removing neutrophils, and she sees that there is a reduction in the protection of anti-OX40 treated animals. And we tried to apply this on something that is approved and known, and we did this with anti-CTLA4 and PD-1 combination, and you see that, in fact, when you deplete neutrophil in this case, you have a reduction in the tumor protection. And so, and then we tried to look in the context of CTLA4 and PD-1 treatment, and we looked at biopsies from patients which after therapy use our own therapy, and you see that in general, so more presence of this netosis or an enrichment of these nets in patients which underwent immune checkpoint blockade. And so, and then trying to convince ourselves even more. So David also did this analysis on this series of patients, which on which an in-depth analysis was done in patients treated with anti-PD-1 only or anti-PD-1 and CTLA-4 combination, and we see that anti-CTLA-4 experienced patient which underwent anti-PD-1 actually had an enrichment of this cluster 1 neutrophil signature in the patient that actually did better. And so suggesting that maybe neutrophils do have, this type of neutrophil or this signature are associated with a better response in given setting in anti-CTLA-4 treated patients. And so we came up with this model, which I think everybody has seen and was published, thinking that, in fact, when you are engaging T cells and in a way that they produce pro-inflammatory cytokines, which are able to either bring in or change the phenotype of neutrophil in a way that they get activated and actually become cytolytic or tumoricidal. And so, but then there was one question that was remaining, right? So we looked at tumor elimination. However, we have seen immune-related adverse event in the animals, and how does that relate? And so, so we wanted to look at these in the context of immune adverse events, and I just want to remind you that at the beginning, what I shown you is that we saw this cauliflower ear phenotype showing up, and again, it's not just in the ear, it's in the ear, snout, it's any non-hairy part of the animal because of where melanocytes are located. They're located in the dermis, so it brings in T cells which are actually recognizing melanocytes there. And so, and we were able to correlate the disease with the presence of ear thickness, and this is how we measured it. And so interestingly, what we saw is that comparable to what we saw in the tumor, there was accumulation of Ly6G positive cells and what we think are neutrophils within the ear of these mice when we under therapy. And similarly to what we have done looking at the tumor, we actually tried to over time, by sacrificing the mice at different time points, looking at infiltration of T cells, which were adoptively transferred, and the timing of the accumulation of the neutrophils in the ear. And you see that as T cells are leaving, it seems that neutrophils are accumulating more within the ear, suggesting that there is a disconnect or these things is like passing the baton, basically. When the cytokines are produced, then they bring in these neutrophils on the tumor side. And what we saw is that actually treating without anti-OX40, get a CTLA-4 and PD-1, this is what I was saying, you'll see why before, we really saw this immune adverse events only when T cells had seen anti-CTLA-4 or anti-OX40. And with anti-OX40, the phenotype was more exacerbated. So I don't know if, we would not see this accumulation of neutrophil with anti-PD-1 or anti-GATR, or our treatment regimen is adapted to that, to these two therapies, which is anti-CTLA-4 and OX40. And what we saw here is that in the model where we only had B16, this is not the mixed model, and when we treat with anti-Ly6G, the ear thickness or the immune adverse event was reduced. However, the tumor protection was not reduced. But of course, if we had heterogeneous tumors, the expectation is that the tumor burden will be affected as well. And so again, here, we looked at nets in the ear, and we saw that there was accumulation of nets within the ear, which correlates with the presence of neutrophils as well, suggesting that this is something similar to what we have seen in the tumors of the mice. And when looking at patients, and this is a patient which was treated with PD-1 and anti-CTLA-4, where you see accumulation of nets as well. And then we looked at multiple patients, and you see that in general, the presence of skin rashes when in the presence of immune checkpoint was associated with accumulation of nets. And so this led us to think that maybe these T cells, which are activated within the tumor microenvironment, would lead to the production of this cytokine when they are going into a different site where there are antigens which are similar. And we were thinking, when we think of oncomimic, for example, we think that maybe this might happen in patients, not just for antigen which are shared by different lineages, but maybe if you think of skin microbiome, maybe skin microbiome is bringing these shared T cells within the tumor microenvironment, within the skin, and maybe, of course, this is huge speculation. And the question we're asking right now is the immune adverse event iNOS dependent or associated with the same signature as the one we've seen in the tumor? And this is something we are trying to answer at this moment, or maybe in this case, netosis is important. And so again, so the model, our working model is, in fact, when you have inflamed skin or you have off-target destruction caused by neutrophil activation, neutrophil infiltration and activation, and this is due to either having shared antigens, or it is mainly due to shared antigen, but and so when the T cells are killed, in the case of the melanoma differentiation antigen, you see depigmentation on vitiligo. However, when you bring in innate cells, you see skin rashes and this type of immune adverse events. And so what we'd like to do in the future is define those differences between pro-tumorigenic and anti-tumorigenic neutrophils, the one similar to what Michaela shows, for example, and others, of course, and characterize the role of these neutrophils and contribution in patient treated with immune-based therapy, and determine the contribution of this neutrophil in immune adverse event, and trying to understand whether this immune adverse event and the bystander killing effect in the tumor we're convinced they're both driven by neutrophils, but is it through the same mechanism? And of course, try to derive biomarker these studies. And finally, I'd like to recognize, of course, people who have contributed a lot in this work. And Daniel Hirschhorn is the main driver in this project. Lucas is a dermatologist who's making a deep dive in this immune adverse event phenomena, and we're trying to understand it. Of course, Jed, long-term partner, and everybody else in the lab and our funding sources, of course. And the people who of course, Michaela's group has had a huge contribution in this project, and shout out to the folks working on immune adverse events, statistician and bioinformatician, who really played an important role in this type of studies, and I'm still trying to understand them, but the bioinformatician, not the studies. I'm kidding. Thank you so much. I'm happy to take questions.
Thank you so much, Taha. That was just phenomenal. There's a number of questions in the chat, so I'm wondering if folks want to turn their camera on and ask the questions. It would be great to see your face for the questions. Otherwise, I can start going through some of them. Nan, go ahead and ask your question since your hand is raised.
I Hi, Taha. Beautiful talk and good to see you. I finally know where I saw you last time.
Okay.
Beautiful
What about-
talk, and
Thank
yeah, we
you.
really enjoyed your paper. And I have a question about your neutrophil gene, your RNA-seq data, or I guess it's single-cell RNA-seq data in cluster
Single-
2.
Yeah.
So you saw you eventually focused on NOS, but in the gene list, you saw a lot of like IL-1 beta, oncostatin M, and a few other inflammatory genes right there in cluster 2. So I wonder if you followed up on some of that, and then what do you think? What roles those genes or those proteins are playing? Because those are typically pro-inflammatory, may not be good in some cases, but I wonder if you can comment on that.
Yeah, so we are trying to follow up on that, and mainly we're thinking on, because with Mario Lacouture looking at, mechanistically we're interested, but therapeutically we're interested too, because we're thinking how to, overcome some of the immune adverse events in the skin or skin rashes, and so blocking cytokines was not always. So you don't know whether it's the experiment that didn't work or it's the biology that's not your friend there. So I think it's really hard to make any conclusive, have conclusive conclusions, if that makes sense. But one thing that we've learned is that when we have tried to reduce that signature and apply it elsewhere, it didn't quite work, right? And so we think we honed in on a flow panel with 13 genes, but we're not sure that this is really working out. So, and the other intriguing thing, we have another setting where neutrophils are also killing, and this is mainly by CD40, like the Lausanne group, CD40 and MPL activation, and this was over another project, and it's interesting because we have tumor elimination by neutrophils, but the neutrophils look different. And so now we're trying to understand what is the intersection of both. And I know there is nothing conclusive, but if I had something, I would have shown. So
got you. Thank you so much. Beautiful.
And I see Santiago ask the question about the CD4s, and so if we are depleting T conventional versus Tregs. So we really, so most of them, with the OX40 treatment, the Tregs will be much lower, right? So I don't think we're depleting, I don't think it's Treg driven. Based on that, we've looked, so the first thing we had looked at initially is looking at Tregs in this system, and we haven't seen that Treg had a role, but maybe, the other thing is the conventional T cells CD4 conventional T cells may come in different flavors.
And Taha, Santiago asks a second question. His internet's unstable, so he's not able to come on, but he has a second question about the early versus late stage and how this might play into some recent work on neutrophils in low versus high grade tumors.
That's a very good point. We have not. So we mainly we didn't do that game with neutrophils, and I think probably we should. We depleted them continuously, right? And because I think it shows. It's not frustration, but we were always afraid we don't deplete enough neutrophils, so we went gangbusters on them, right? So we did not. But it's a good point. I like your suggestion.
So I have a question. Have you observed or have you looked at any effects on monocytes? Do you also see recruitment of pro-inflammatory monocytes?
We see high infiltration of macrophages, but they don't seem to play any role, at least in the bystander killing effect. I don't know if this answers your question.
Yeah.
We have other questions from Joanna Joyce.
Taha, that was a lovely presentation. Thank you very much. I have a number of questions. Some of them are technical and some of them are more kind of conceptual. Maybe technical first. You mentioned about you went gangbusters to try and deplete the neutrophils. How long were you able to do that for? Because I think that's maybe an important point to discuss here in this forum, right? The challenges with effectively depleting neutrophils and not getting this rebound, massive egress of neutrophils from the periphery, right? So maybe you could just let us know how long you were really able to effectively see neutrophil depletion in your animal models.
So my recollection is that, and if Daniel was here, that would be better. But we went until day 20.
I'm here.
Yeah, okay, Daniel is here. So Daniel, can you answer that? How long?
Yeah. We depleted for close to 3 weeks. We were doing 500 milligrams of 500 micrograms of the antibody every other day. So the other thing is that the mice receive cyclophosphamide. So that kind of set up different. That sort of changed a little bit the environment itself. I get that question all the time because apparently depleting neutrophil is tricky, and the reason we had to go gangbusters is because of this idea of introducing cyclophosphamide, changing the environment, and then repeatedly dosing with the antibody until we see some effect. I mean, in some of the depletion experiments, we did it at several time points, and the depletion worked really, really well.
I think it's a really important point to bring up in the field, and I see this comment from Amy Jo, and you have your camera too.
And I can tell you, Joanna, I remember I went to the neutro meeting where we spoke about neutrophils for 2 days, and people were very curious about this, and this question was asked again and again, and I think the agreement at the end was that yes, you are able to, if you give enough anti-LOS6G in the B6 background, at least you are able to block or deplete neutrophils to a certain extent, and it is efficacious.
Yeah, maybe it depends on the model system.
It's temporary,
right?
yeah. I guess that's what I wanted. The depletion is really temporary, and I think it's also just due to the antibody. I don't know if it develops ADA, or I know there's several papers about the specific mechanism of why the Ly6G doesn't work after many times, and so I've used it in the past too. And maybe the first couple of weeks you can see depletion, but after that they just come back, with great vengeance.
And I don't think we are going beyond the first couple of weeks.
Yeah.
But this question haunted us, right? So we checked. I mean, this was like the recurrent question.
Yeah, because I think it's like a lot of things in the literature. If it's not reported, right, then if the perceived dogma in the literature is that these strategies work, and then we try them in our labs and they don't work as simply as has been reported, it becomes really difficult then, right?
Yes.
Because
but I think,
we're trying to do the experiments, we're trying to respond to reviewers.
Yeah.
This has been shown by X, Y, and Z to work, and then it doesn't. Again, it can be model dependent. Of course, it's definitely antibody dependent. But I think maybe it's something that we can see as a community, right? Given that this is a myeloid cell network, how to kind of how to deal with this. Anyway,
I think we need to remember the point that Daniel raised, which is quite important. We are in the context of giving quite a high dose of cyclophosphamide where we induce lymphodepletion, and so we're changing, the different immune cells are not repopulating at the same time. So
yeah. I don't want to monopolize the discussion, but I did want to ask 2 quick questions, if I may. Did you show that nets were critical for some of the phenotypes that you observed? And the second question, you had this nice slide, neutrophils are confusing, I think is what you said. it is confusing, right? Because in different contexts, they literally have opposing roles. So that was the more philosophical point I wanted to get your take on. If you have any consensus on this, or it's like what we were just talking about, the depletion. It depends on the model, it depends on the cancer type, it depends on the strategies, and we all need to do it in our own hands and see the data that we get. And neutrophils are indeed very confusing, very complex.
Yeah. So one thing that of course we're thinking about this, and I remember even when I wrote at the beginning the discussion with I'm with Colombo, with Picala, and because we're like we are part of two different clubs. Neutrophils are killing, no, neutrophils don't kill, or proteomorigenic or anti-tumorigenic. And I remember it's funny because at the end of writing a grant together, she goes, Taha, now I buy it. But maybe the context within the tumor is important, right? So you see how we show that the neutrophil come after the T cells are gone somehow, right? And so maybe, and this is pure speculation, maybe neutrophils, when they're present at the same time as your T cells, maybe they don't eliminate only tumors, maybe they eliminate effector cells, or maybe they have a negative effect on the effector portion of the response. So that is a possible interpretation. I don't know if it's the interpretation, but I think if we not apply this exactly as an example, but this is how I'm thinking that maybe context is different. But also maybe it's simply that those are different phenotype or signatures. This is why I really want to take in the same lab, in the same setting, doing this, looking at proteomorigenic versus anti-tumorigenic neutrophils and compare them side by side in the same experiment. And I think maybe we'll learn something there or get more confused. I don't know.
Okay, thank you.
Amy Jo, I think your question wasn't asked or answered yet, right? About the systemic neutrophil expansion versus local.
Yeah, I was just curious if when you see that neutrophil expansion in the tissues after treatment, if you see also systemic expansion, if you also see, massive bone marrow production of neutrophils, and if you see an increase in the blood too, or if it's very a localized recruitment versus expansion.
Daniel, you want to take this one with more in-depth?
Yes, we looked in other tissues, and we only see it in the tumor.
And so you don't see it in the blood?
Although I do no, we don't see it in the blood, although I never checked the bone marrow.
Okay. Yeah, you would see it in the blood if it was increasing in the bone marrow, because it would come from the bone marrow. Okay. Thank you.
Sure.
Hi, Ding. Do you want to ask a question?
Yeah, hi. So it is very interesting talk. Thank you very much. And we also discussed our paper in Journal Club together with other paper from Michael Pittay lab.
Yes.
So can you comment on how similar the anti-tumoral neutrophil that you found in your single cell analysis data with the interferon stimulated subset that they found? And is it possible to sort those anti-tumoral neutrophil out? Is there any survey marker that you can sort them out and study them?
Sort them, enrich for them together with other cells, yes. Sort them out. I think the Pitet neutrophils and ours, in terms of signature, maybe a little different. And again, what I was alluding to is we have another story in the lab where we see neutrophils which are tumoricidal and they do look different in terms of the signature, and we have been scratching our head as to why they come in different flavors.
Yeah, and the reason I add is we see something similar in our model, but and then I'm wondering, like, because it related to the depleting, I'm just wondering, like, when we deplete them, we also deplete anti-tumor neutrophil. And that is maybe the reason that the anti-Ly6Z depletion didn't work well as we expected. I don't know, that's just a thought.
So that you're depleting anti-tumor neutrophils.
Yeah.
But that's what we believe we're doing. The thing is, I think, depending on
we deplete both, right?
But depending on what's more enriched in your model is what you're going. That's what your readout will be. Yeah. Right. So we're fooling ourselves. I mean, I don't know. Maybe I'm too simplistic.
Yeah, and also like the recent paper from Edgar Engelmann lab when they activating neutrophil using TNF, anti-CD40, and tumor antibody, and they make neutrophil killing better. So it's just like, I don't know, like it's very confused. Like when should we deplete? When should we activate? I think that's probably going to be the question for the whole field.
But the other question, right? And so these anti-tumorigenic neutrophil, do they all kill the same way?
That's true. Yeah. I don't have.
So we are assuming they all kill the same manner, but now more and more I believe it's not.
No, it's not. I agree. Yeah, it can use T cell or it can use non-T cells mechanisms. Yeah, great talk. Thank you.
I had a question. I wanted to ask a quick question, Taha. So you showed that as T cells are waning in the tumor, the neutrophils are rising in content. But did you look at subsets of T cells? Is it possible that the neutrophil recruitment is associated with a particular subset that's not declining?
So we are doing adaptive T cells. So we did this in the setting where we did it both in immune competent and in rags. And so we did it in the presence of only our CD4s, right? So this is the artificial but also cool thing about adaptive T cell transfer, right? So you know that you don't have any other cells left. But I agree. And initially, this is how we were thinking about it, right? We were always thinking that it's the other T cells are taking the relay. Because, I mean, we like myeloid cells as suppressive, but not killing. And then the data always took us in a different direction.
Okay, thanks.
Taha, there's another question from Payal Yokota asking if the neutrophils are predominantly coming from the gamma delta T IL-17 axis, is it possible to do a combinatorial blockade to efficiently keep neutrophils down? And Payal, I don't know if you're still there.
Yeah. Hi, Taha. Oh my God, it's been a long time. I was wondering.
Hi.
Sorry, I have to know again without a question.
Yeah, I don't know how many gamma delta T cells we have enriched there. In our adaptively transferred T cells.
I see. Okay, yeah.
But it's certainly something to look at. I don't think it will be, because we are really biased to this, I call them super killer T cells, these CD4s, which have a weird phenotype, but they are highly cytolytic, right? But they, again, maybe in the context of the MC38, where we don't have adaptively transferred T cells, we might look at that.
Great, thank you so much. Lovely talk.
Thank you.
Are there other questions? I don't see any others in the chat. As others might come on to ask questions, there's a place on our website where we have a section for protocols or shared resources. I'm wondering if we can take some of these papers that were either sent in the chat or mentioned and put them together on the web page. So maybe we can work on that and make this a resource for others. And if anyone has anything to add, if they could email it to the Myeloid Network website or email.
So one suggestion, which may be crazy and not very practical, I think it would be nice if we actually, even these technical questions of neutrophil depletion, is is having 3 or 4 different labs, try something in parallel and actually really compare notes and then getting to the bottom of it, right? I think that would be very, very helpful. It's not going to be scientifically earth-shattering, but I think it will help all of us, and we will be more confident into the result we are producing. I propose that.
Love it.
Yeah, I think that would be a very nice thing to do, and I think that there would be several groups that would be interested in that, including our group.
Awesome. Yeah, I agree.
Can I make a quick comment? From some discussions I had with some neutrophil people, there's this mouse model that's called CEBP epsilon knockout mice. There's a story about that's the master transcription factor controlling neutrophil maturation. I don't know. I'm also new to this. This feels like a lot of mature neutrophils or activated neutrophils rely on CEBP epsilon to be activated. So that's another model I've heard of, as a way to, it's not a depletion model, but it's a deficiency model. The GCSF model knockout, I don't think it works if you look into bone marrow neutrophil numbers, precursor or whatever. But anyway, just a thought. And I agree with Taha, we should work on this together. And great talk. Thank you.
Yeah, the other point about all of these neutrophil depletion experiments and all that's been reported in the literature, if it's not necessarily in the context of cancer,
yes,
it can be very different, right?
Yes, absolutely.
And so I think there's probably amongst us here people who are interested in manipulating neutrophils in different contexts, not just in cancer. And so we also need to be very cognizant of that. Cancer versus not cancer, different types of cancer models, because they're all having an impact on the neutrophils and vice versa in really quite different ways.
Yeah, perfect.
Okay.
Yeah, I think that's fantastic. I think we should get together, and actually we should all be coming together at the SITC annual meeting next month. So hopefully we'll see you all there. There's no Myeloid Network seminar because we're hosting the pre-conference workshop at the on myeloid cells at the SITC annual meeting the first week of November. It's on our website. Hopefully we'll see you there. I think more to follow on this really fantastic idea to come together as a community and tease out some of these important questions related to protocols in both cancer and non-cancer. So we'll plan to reach back out to the community with more ideas on that. So thanks so much. And we'll hopefully see you at SITC next month. And thank you so much, Taha, for such a great talk and a wonderful discussion.
Thank you. I really enjoyed it. Thank you so much.
Thank you, Taha.